Okay, jumping back in. I'm going to start lecture 4 talking about enzymes. So defining what an enzyme is, it is a substance produced by a living organism which acts as a catalyst to bring about a specific biochemical reaction. Main thing to focus on here is that it's a catalyst. It makes the reaction happen faster. It does this by reducing the activation energy. Another big component of an enzyme is that it is not used up during the reaction. So you should have the enzyme at the beginning and the end of the reaction. If you don't, then it's not an actionable enzyme. Okay, so what is the effect of enzymes on the reaction rate? It speeds up the reaction rate. The effect of enzymes on activation energy, it decreases the activation energy required. There's still some energy required, but it decreases that energy. We also have cofactors and coenzymes. These are used to activate the enzymes. It binds to what we call the apoenzyme, or the inactive enzyme, to make it a hollow enzyme, which is the active form. Yeah. Okay, so we talk about cofactors and coenzymes specifically. So cofactors, a little fun mnemonic to remember that. Think factor, think fear factor. You have to be pretty metal to be on fear factor. These are going to be our metals, our iron, magnesium, manganese, zinc, copper, potassium, sodium, and spacing what that is right now. That's fine. These one's a good example. Iron, magnesium is a big one, so remember that one as well. And potassium, sodium. Coenzymes, these are going to be our vitamins, our complex organic molecules. So if you remember, we talked about all the B vitamins. Remember, tall, rich, nudists, played pickleball for centuries. All the different B vitamins: thiamine, riboflavin, niacin. I get the P's mixed up. One is the pyridoxine. The other is the, spacing of the name, B, biotin, folate, and calmodulin. Alright, there's pyridoxine and pantothenic acid. Those are the two B's. There we go. Yeah. Remember your B vitamins. These will come back a lot on boards, as well as in a handful of classes. General pathology, systems pathology, a little bit in physiology, as well as clinical nutrition. Your vitamins, especially your B vitamins, are going to pop back up. Lab di as well. Your lab diagnosis class, that is Tri 5, that will pop up there as well. So you'll be seeing a lot of these. So please take note and remember those. The different enzyme classifications. We should have a lot of practice seeing these different names of enzymes. So the oxidoreductase, what do we think it does? Oxidation reduction reactions. Transferase, it transfers a group. Hydrolase, hydrolysis is breaking apart something. So the peptide bond, for example, breaking it down, hydrolyzing it with water. Lyase is again breaking bonds, but something that doesn't use water. Isomerase, it's turning into a different isomeric form, so kind of rearranging it. And a ligase is building bonds, creating bonds, putting this stuff together. What is the active site of an enzyme? That is where the substrate binds for the reaction. What is meant by substrate specificity of an enzyme? Each enzyme will bind to one specific substrate. So 1-to-1. Each enzyme is very specific. In general, the enzyme-substrate interaction, so the substrate binds to the enzyme, forming the enzyme-substrate complex, and intermediate joins and products are formed. The intermediate can be, you think about the, you also need the coenzyme or the cofactor. You have different reactants that then form together with the enzyme to form products. So we're talking about transition states now. This is just showing that activation energy that we need for the reaction to occur. And so this is our normal, our ground state. Then we need to get all the way up over this hump in order to go from substrates to products. The purpose of an enzyme is to have us from this amount of space, and it's going to literally cut the amount of energy that we need. It can be by half or even less. So that way we need a much less energy for this reaction to occur. Catalytic strategies, acid-base catalysis. Active sites of some enzymes contain acidic or basic functional groups that act as proton donors or proton acceptors respectively and interact with the substrate. You can have covalent catalysts or metal ion catalysis as well. The effects of the following on enzymatic activities. Go ahead and pause and take a second real quick to read through these. These different things will have an effect on how the enzymes work. Basically, there's an optimal pH, an optimal temperature, an optimal concentration. There are also activators and inhibitors, which activate the enzymes or prevent the enzymes from doing their functions as well. And all of this is for, maintaining the body's homeostasis, making sure that the right things are happening at the right times. Talking about inhibitors, we have 3 different types. We have competitive, which directly binds to the enzyme substrate binding site and blocks the substrate. When this happens, the Vmax, so that's the velocity at which the reaction occurs, remains unchanged, but the Km increases, which is the amount of time that it's going to take it to get to half of its Vmax. You have uncompetitive, where the inhibitor is binding at a separate site of the enzyme, and that decreases the Vmax and the Km. Mixed or non-competitive, it binds at a separate site but may bind to either the free enzyme or the enzyme substrate complex. Vmax decreases but the Km remains unchanged. These as well, these will be board questions, these guys, so it's really just kind of memorizing these couple of details. The Vmax, what happens to the Vmax and the Km with each of these. The Michaelis-Menten equation of enzymatic kinetics. So the rate equation for a one substrate enzyme catalyzed reaction. So this equation relates the initial reaction rate, or V naught, the maximum reaction rate, or Vmax, and the initial substrate concentration. The Michaelis constant and its significance, that's the Km. Km is the substrate concentration at half of the Vmax. And the substrate concentration at which the reaction velocity is 50% of the Vmax. So how that reaction is occurring at 50% of its maximum capacity at that point in time. Irreversible inhibition of enzyme activities, stuff like heavy metal poisoning. This is like certain inhibitors are going to bind and never release. So literally the body they cannot use them. So some are irreversible, some are reversible. It just depends on what's binding. No one be able to describe allosteric effect on enzymatic activities. So the allosteric effect. This is the binding of a ligand to one site on a protein molecule in such a way that the properties of another site on the same protein are affected. So hemoglobin is like this. So if the first oxygen binds to the hemoglobin, to the T hemoglobin specifically, it's going to increase the affinity for the binding of oxygen in the other parts. So it's saying, an allosteric effect, it binds on one part of the protein, and another part of the protein is affected, and there's a response as a result of that. These right here, cell signaling, G protein and protein kinase A. These videos, you don't have to go back and watch the specific podcasts. if you don't want to, but I would recommend at least pulling up, especially with the G protein coupled reactions. Make sure you really understand that portion. It's a process that will come back as well in a couple of future classes, as well as cell signaling will come back in a handful of classes. So these topics will not go away. Please remember these because you will need them for the future as well. But kind of talking about cell signaling, so we have cell-to-cell communication. First off, there's a couple different types of cell, the way that the cells communicate. There's an autocrine. So the cell is going to produce a signal. Typically, when we say it produces a signal, it's going to be releasing some sort of ligand, some sort of chemical that's then going to go and bind to receptors and cause something to occur. Cause like a process or something to happen. So autocrine, a cell is going to produce a signal that will activate its own receptor. So the cell produces a signal, then that signal then acts on the same cell. Paracrine goes short distance cell signaling, so it's going to affect the cells directly around it. And then you have endocrine, which are long distance cell signaling. It's going to go into the bloodstream and travel all over to other cells throughout the body. Those are our hormones. The endocrine system is our hormone system. When we talk about these receptors, so that these ligands, these signals, are going to go and they're going to bind to receptors. These receptors are specific, meaning they will only bind to that specific ligand. They have high affinity for their specific ligands, which means that when they're given the opportunity to bind, they really want to bind. Most often, these are also going to be reversible, unless like something binds into it that makes it irreversible, something that's not supposed to be binding there, like those heavy metals or something like that. Saturable means that you can have a concentration where no more reaction is occurring, so all of the receptors can be filled. Ligands bind to the receptors, it integrates and amplifies the signal, produces a biological response. This is the whole point of these signals, is to produce a biological response. And it will be terminated after that response is complete. So it can be turned on and it can be turned off. When we talk about these receptors, there can be desensitization and adaptation to it. So if we have a ton of ligands, a very high ligand concentration may cause desensitization. And we call this down-regulating. So those receptors, they get internalized by endocytosis. Basically, the cell is going to take the receptors and kind of like grab them and suck them back down in through the membrane, so that they're no longer available because they're getting stimulated way too much. Kind of think of they're turning on their introvertedness and just kind of hiding, basically. This is mostly seen specifically when we talk about diabetes, specifically diabetes mellitus type 2. With that, basically the body releases insulin over and over and over and over and over again, and so much insulin that the cells become desensitized to the insulin. And interestingly, the solution to that is actually to literally inject yourself with more insulin. So you can kind of see how that becomes a vicious cycle, where the longer that you stay on insulin, the worse off you actually are with type 2 diabetes. There is, though, you can have the opposite, which is upregulation, which is an increase in the number of receptors on the surface of the target cell. And just depending on how what you do with your body, that you can reverse a lot of the time. Maybe not fully, but you typically can reverse some of the downregulation that has occurred. So there are different types of chemical messengers. When we talk about the nervous system, there's a whole bunch of these. You need to know these for physiology, physical diagnosis, systems pathology, general pathology, and through several other classes. Phys 1, you'll hit a lot of these hard as well. So get familiar with these now, so that way you're a little bit set up for the future classes. So the neurotransmitters in the nervous system are acetylcholine, norepinephrine, epinephrine, and GABA. Those are the big ones. There's a couple of others, but these are the main ones that we focus on. We also have the endocrine system. We have the hormones. We have the steroid hormones, such as testosterone, estrogen, and cortisol. We have thyroid hormones, norepinephrine, and epinephrine as well can act as hormones. So, or norepinephrine and epinephrine are pretty versatile in how they're used. It just depends on where they are released. So in the nervous system, they're released in between from neurotransmitters from the nerves, the synapse. And then directly into other nerves. In the endocrine system, they're released into the bloodstream. The immune system also is going to have chemical messengers such as cytokines, eicosanoids, and histamine. These right here, huge part of biochem too. Don't worry about them now, but these will come back next trimester. Our intracellular receptors. So this is, we need to remember these. There are different types of receptors, or different locations for these receptors. We have cytosolic receptors, we have nuclear receptors, and we also have cell surface receptors, okay? So there are receptors found within the cytosol, there are nuclear receptors. Remember this list here, the steroids, the vitamin D, the retinoid hormone, and thyroid hormone, specifically steroids and the thyroid hormone, are the big ones that you'll be asked about. It means that they're able to go through the membrane, the outermost membrane of the cell, and they go directly into the actual nucleus, where they then bind onto the nuclear receptors, which are going to have a direct impact on the DNA and the expression of genes in the DNA. We also have our cell surface receptors, such as getting into our G protein coupled receptors and our ion channels. So we're going to talk quickly about hormone action, and then eventually later on we'll get into some of the G protein coupled receptors. So mechanism of hormones action. So it's via membrane receptors. So we have the peptide hormones such as insulin and glucagon. We have amine hormones. They produce secondary messenger molecules. And they cause action through activation of one or more nearby functional proteins in the membrane or the cytosol. And there's a stepwise cascade pathway, which leads to a biological function or response. And these are faster action than steroid or thyroid hormones or vitamin D. So they bind onto the membrane receptors, so there's receptors just on the outside, and it causes a cascade reaction. So it's kind of like there's one reaction here, and then that one reaction sends two signals, and then each of those signals send two more, and each of those send two more, kind of like a pyramid, and it cascades down through, causing a very, very quick reaction to occur. There are also those nuclear receptors we talked about. These cause specific gene expression because they literally go right to the DNA, and these tend to be a little bit slower action than some of the other hormones. Here's our chart with all of the hormones on it. This is in the book as well. Recommend reviewing those and where they're found and where they're located, all that sort of good stuff. We have some second messengers, the cyclic AMP, the cyclic GMP, calcium, the IP3, etc. A lot of these are mostly the cyclic AMP we'll talk about when we talk about the G protein coupled receptors. Okay, let's look at the picture as a whole. Just going to fly through this really quickly. If you need another review, you can go back and watch the video that I have about this entire reaction, or you can always just look something else up. Look up the G protein coupled receptor reaction on YouTube as well. Okay, so we have several different components of our G protein. We have the alpha, the beta, and the gamma. So the alpha, the beta, and the gamma components. And it has a GDP bound to it. A GTP is going to come in and activate it. When the GTP binds, the GDP pops off, and we have the active form. The active form, the beta and the gamma subunits are going to separate from the alpha and the GTP. And now this is going to act on our adenylate cyclase. The adenylate cyclase then, through addition of ATP, gets converted to cyclic AMP, which is our secondary messenger molecule. This is going to go and act on our PKA, our protein kinase A. And protein kinase A, remember, is in its inactive form. It has 4 subunits, 2R and 2C. Make sure that I get all of this right. The cyclic AMP is going to bind and remove the R subunits, and our 2C subunits are left by themselves, and that is then the active form of our PKA. Addition of a phosphate in the substrate, so that's some sort of protein, you get a phosphorylated protein, and this phosphorylated protein is then going to kickstart the reaction inside of the cell to continue and carry on the signal and bring about the functional change that we're looking for. And then, of course, the CAMP gets deactivated, and the adenylate cyclase also gets hydrolyzed by the intrinsic GTPase, and everything goes back to its original start position, waiting for another ligand to come and bind. So here's all of the different words specifically for that, kind of spelled out. Again, this is in your book, spelled out word step by step. I'd highly recommend going back through this and reviewing if you were a little bit confused or don't recall this reaction. Okay, let's see what else we got. Okay, we talk about phosphodiesterase, which breaks phosphodiester bonds. Again, just kind of vocab to remember. Here's all of this again. This is again going through the components of the cellular signaling pathway. Again, this is the exact same pathway over and over and over. It's kind of listed a couple of times, so it's pretty important. Characteristics to be a receptor molecule. This is again a repeat of above. Some examples of the G protein coupled receptors. We have adrenergic receptors such as norepinephrine, epinephrine, and dopamine. The acetylcholine muscarinic receptors, GABA receptors, and glucagon receptors are some of those G protein coupled receptors. We see our secondary messenger molecules again, which are listed above as well. The mechanism of GPCR, ligands bind to the membrane receptors, signals transmitted to G proteins, have 3 subunits. And this is again more of the same. This whole part of his review kind of was very repetitive as I was filling it out. So, okay, so hydrolysis of GTP will release energy. Again, this is still going into more of the details through the G protein coupled receptors, which we just talked about. There's a lot of words here. Don't worry so much about these words. If you understand the process behind it, you'll be fine. This was more studying for me. I did this personally. I was looking through G protein coupled, trying to understand a little bit better. Okay. Lecture 6, regulation of fuel metabolism. So So the two general mechanisms of the hormone action, we have membrane receptors, nuclear receptors. Hey, look, all this stuff is here again. We just talked about it. This is the exact same thing that we just said above. We have the membrane receptors, we have the nuclear receptors, those different types. Remember that chart up here. Remember your different types of your hormones and stuff to remember. Very repetitive. Okay. Our main energy molecules, we have proteins, carbohydrates, and lipids. All of these will be broken down to produce acetyl-CoA. And now at this point, we should know that acetyl-CoA goes where? Acetyl-CoA goes into the Krebs cycle to produce our NADH and our FADH2 in order to go to the electron transport chain to produce ATP. Insulin and glucagon, they work opposing each other. So insulin is going to decrease blood glucose levels, and glucagon increases blood glucose levels. So think about when each of those are going to be released, why they would be released, and kind of what effect they would have on the body. Which is what we just said right here, the changing in the blood glucose levels. Cortisol versus epinephrine. So epinephrine is adrenaline. It's a quick-acting rush of glucose to prepare the body for action. So our fight or flight response will give us a shot of adrenaline or epinephrine. Cortisol is more of like a long-term adjustment to maintain the glucose levels. And too much cortisol decreases immune function. This is one of the reasons why it's hypothesized why you get sick after really stressful times, like after finals. Most people will have a day or so where they just feel really, really run down and ragged and they get sick because especially during practicals and exams and everything, our cortisol levels tend to spike because it is known as our stress hormone. And that is what we will see. Making of insulin. These are the steps. It starts as pre-proinsulin. Then it gets cleaved and makes proinsulin, and then we will end up with active insulin by another removing of this C peptide. I would know the names and just the rough, like there's cleavage of stuff off, like the C peptide. I would know that. And then the active insulin can go and bind to its receptors. The different effects of insulin on the blood glucose, where you get increased glucose uptake, increased glycogen synthesis in the liver and the muscle, decreased glycogen breakdown, increased glycolysis, increased fatty acid synthesis, and increased triglyceride synthesis in adipocytes. And then all of these are opposite. The arrows would be flipped for glucagon. The mechanism of action of insulin via its membrane receptor. So the insulin receptor is a tyrosine kinase receptor. And don't worry too much about this. Understand that it's a tyrosine kinase receptor. That's like the main point. You'll get a lot more of this in Phys 2. There's a whole section dedicated to the tyrosine kinase receptor and insulin and glucagon. So, but yeah, understanding that the interior side of the tyrosine kinase kind of has like these branches that are out, and those will be phosphorylated when insulin binds, and that phosphorylation will cause a cascade response on the inside of the cell, promoting the GLUT4 to bring glucose into the cells. The effects of glucagon on blood glucose, again, it's going to be opposite of the insulin. The glucagon receptor is a G protein coupled receptor, and we talked all about those just a minute ago. So that's the pathway it's going to follow. These specific hormones or the metabolic homeostasis hormones, there's a handful of them. The insulin and the glucagon come from the pancreas, specifically the insulin from the beta cells and the glucagon from the alpha cells. We also have adrenal hormones, the epinephrine, norepinephrine, and cortisol releasing from the adrenal hormones. It's not part of the kidney, it actually sits on top of the kidney. And there's one on top of each kidney. You'll get to see those in your dissection labs. Try 3 is when you do a lot of the organ systems. Here's relating insulin and glucagon, how they have negative feedback. So as we can all through is when insulin does its job and brings the blood, decreases the blood glucose, it feeds back and says, hey, stop, we don't need any more insulin, good to go. Same thing with glucose, or glucagon. So the regulation of blood glucose by insulin and glucagon, this is saying pretty much the exact same thing. There's a lot of repetition here. Sit down and write down what the function of insulin and what the function of glucagon is, and if you understand that well, you'll be pretty set for this whole chunk. Again, saying where they are created. So the role of epinephrine and cortisol, we already went over that. The effects of high blood glucose on the synthesis, secretion, and action of insulin in glucose transport. This is a little bit more specific than we need to go into it, so don't worry about that. And look at metabolic action of insulin in several tissues, particularly in liver, muscle, and adipose tissue. So again, this is literally just the same lists. It's just he kept asking the same exact questions in different ways. Then how mature insulin is formed. We already talked about that. The mechanism of action of insulin via its membrane receptors, we talked about that as well. I think this might be a little bit, this might have gotten duplicated a little bit as well. Yeah, a lot of this has been repeated a couple of times, so that should be good. I think that horse has been beaten enough at this point. Okay, and that takes us to Lecture 7, Bioenergetics. I think I'm going to go ahead and stop this video here and keep these between 30 and 40 minutes. And I'll try and get the next part up as soon as I can. Keep crushing it. I will see you in the next one.